Okay, so talking about the urinary system, which we're also going to be covering acid-base balance in this lecture because thinking back to the beginning of the semester when we first talked about acids, bases, and pH scale, we learned that there were two major systems most involved with maintaining your body and blood pH. The respiratory system that we talked about just a little bit ago, which will be tested on next week, and then this system, the urinary system. That'll be stuff we kind of get to at the tail end of the lecture. We'll kind of focus in on what the urinary system does aside from that for the majority of the lecture though. And there's a lot, as you can see, that the urinary system does. But what you see listed here as far as functions go truly is strictly related to the kidneys. And those are not the only organs of the respiratory or the urinary system. Sorry, no, it's kind of small text because there's a lot of information here. Your kidneys are the primary organs of the urinary system, and they are what do all these things. Then you have the ureters, there's two of those, a urinary bladder, and then your urethra. We'll kind of just touch briefly on those because there's not too much physiologically that they do. In the case of the ureters, they just carry urine to the bladder from the kidneys. The bladder holds the urine until it's time to pee, and then when you pee, you use your urethra to get that urine out. So most of what we'll talk about in this lecture relates to the kidneys. And functions of the kidneys include the regulation of our blood ionic composition. So keeping the ions like sodium, potassium, calcium, chloride, and hydrogen phosphate in check or in correct balance. Regulating blood pH through the excretion of variable amounts of hydrogen ions, or in some cases conserving bicarbonate ions, which are valuable blood buffers of hydrogen ions. Regulation of blood volume, which refers to the amount of blood that we have, which is easily done by just conserving or eliminating water and urine. And if we remember back just a couple weeks ago when we talked about blood, we learned about half of your blood is just strictly water. So by either peeing out more or less water, that's going to affect your blood volume. And as that occurs, and we've talked about this before, any increase in blood volume is going to trigger an increase in blood pressure, whereas a decrease in blood volume decreases blood pressure, which helps me transition to the next function. Regulation of blood pressure is through those mechanisms, but it's also through the release of an enzyme called renin, which activates the renin-angiotensin-aldosterone mechanism or pathway that ultimately leads to increases in blood pressure. We've talked briefly previously about the renin-angiotensin-aldosterone pathway before. We're going to get into it again here in this lecture this afternoon. Our kidneys also help maintain normal blood osmolarity. If you don't remember, osmolarity is the measure of total number of dissolved particles per liter of solution. So I always just tell students, think of osmolarity as like water balance. So keeping the water levels and the stuff dissolved in the water kind of perfectly in balance. Okay. And so by separately regulating water and the loss of solutes in our urine helps to maintain this osmolarity. Production of hormones, and we've talked about these before, but calcitriol, which we learned is going to help regulate calcium homeostasis, and erythropoietin, which stimulates the production of red blood cells. Regulation of blood glucose levels through the process of gluconeogenesis, which is something we talked a lot about the other day on our cell metabolism lecture. Gluconeogenesis, remember, is the formation of glucose molecules from non-carbohydrate sources. In this case, it would be the amino acid glutamine. Okay. We can also use our kidneys to release glucose in our blood to help maintain blood glucose levels. And then the excretion of waste from metabolic reactions and foreign substances, which include things like drugs and toxins. And these wastes in the body can include ammonia and urea, which we talked a little bit about the other day. Happened to form those when we deaminate amino acids. We also have other waste products like bilirubin, creatinine, uric acid, and then you've got drugs and environmental toxins. Let's talk a little bit more about wastes. A waste is any substance useless to the body or present in excess of our body's needs. So either we have no job for it, Therefore, there's no reason to have it in us, or we just have too much of it. We've talked a lot about some waste here lately. Carbon dioxide, we talked about it being a waste product that we breathe off. We don't pee off carbon dioxide, but we breathe it off. Water can be not only a nutrient that's beneficial to us, but also can be harmful to us, which means it can be a waste product. But when we think about waste, more than likely what we're thinking about are metabolic wastes, which are those produced by the body, like the 4 that are shown over there on the right, which all happen to be a type of what we call nitrogenous waste, which are toxic metabolic wastes that contain nitrogen. Examples of nitrogenous waste include urea, which is a byproduct from the breakdown of protein, the protein catabolism stuff that we talked about the other day. Remember, that's what we form when we convert ammonia to its less toxic form, which is what urea is. Uric acid is a byproduct from the breakdown of nucleic acids. This, if not removed from the body, can lead to things like gout. I'm sure you've heard of gout before. And then creatinine is produced from the catabolism of creatine phosphate, which, if you don't remember, creatine phosphate is one of those energy substrates that our muscles use to make ATP. And as a byproduct, we're left with creatinine. If your body fails to remove its nitrogenous waste, that can be very, very detrimental to your health. A test used to measure the levels of nitrogenous waste in your blood is expressed as your blood urea nitrogen, or BUN. And like I said, if you have an inability to remove your waste, that can be detrimental. And we call this condition azotemia, where there's an abnormally elevated levels of BUN or elevated BUN, which may indicate renal insufficiency. So the kidneys are not properly working, which might lead to the need to perform what's shown here on the right, the artificial removal of nitrogenous waste in what's called hemodialysis, or what most people will just refer to as dialysis. And I'm sure you have heard that term before as well. This is what people have to have done when they have kidney failure. It is a very invasive, fun type of condition or test, or I shouldn't even say test, it's a treatment, where they stick, as you can see, 2 lines into your body, one that draws blood out of your body, and it runs it through this machine, which is essentially going to mimic what your kidneys do, which is the formation or the filtration, excuse me, of your blood, cleaning things up, removing the bad stuff, and then returning that clean blood back to the body. Okay. And I say that it's not fun. Obviously, getting needles stuck in your arm is not a fun thing, but it's not something that it can happen just once and you're fine. If your kidneys aren't functioning, you're going to have to perform dialysis very, very often, okay? A couple of times a week. And it's not a short, slow procedure. It takes hours to get this done. So your job and your life at this point basically becomes dialysis and don't die. And because you can't survive very long without this. And that's what makes it very unpleasant. Really, mental health drastically declines as a result of this, on top of your physical health issues. So obviously the goal would be a short-term dialysis, while hope is you get a kidney transplant, so your body doesn't have to do or your body gets to do the filtering instead of artificially filtering. All right, what we're going to do is just kind of introduce the anatomical parts of the kidneys that do the filtering of our blood if our kidneys are functioning correctly. These structures are called nephrons, and each kidney contains over 1 million of them. And they each consist of 2 principal parts, a renal corpuscle and a renal tubule. Okay. The renal corpuscle is shown here on the left side of the figure. This is a very simplistic illustration of a nephron. You'll see a much more complicated one here in a little bit. Well, this is where we first filter the blood and form the precursor to what we will call urine. And that precursor to the urine is a fluid called filtrate. It's predominantly water, just like any other fluid in our body. And then it's got a lot of other stuff in it, okay, which then flows out of the corpuscle into this long winding tubule where we kind of process that filtrate. We give some stuff back to the blood. We return that stuff so that it is not lost in urine. We push stuff out of the blood and into the tubule. And eventually what we are formed at the end, what we have formed at the end is urine that then drains out through what we call a collecting duct. And we'll get into all these things here as we roll through. I just want to kind of introduce the anatomical parts so that we can understand their physiology, just like we always do. Okay, introduce structure so we can talk function. As I just mentioned, ring corpuscle is where we first filter our blood, forming filtrate. Consists of kind of three major types of blood vessels. Okay, an afferent arterial, which is a blood vessel that brings blood into the entire structure. The entire structure is the corpuscle, and it's going to include not only these blood vessels, but just a kind of a capsule on the outside that kind of encases everything, okay, and collects the filtrate inside of the space. Okay, so the blood comes in. This is whole blood, so it's still got all the stuff that it would normally have. And that afferent arteriole then transforms into a mass of capillaries called glomerulus. Okay, this glomerulus is hard to see because of the fact that there are these cells surrounding the capillaries that are inside. These cells are called podocytes, and their job is to kind of just keep the big stuff in the blood. Okay, they're going to keep the red blood cells, the white blood cells, your platelets, your plasma proteins like fibrinogen and albumin from escaping the blood. We don't want those things leaving the blood. We want them to stay inside. Okay. Well, that leaves the very small stuff that can force themselves out and into this capsular space. And that includes water, urea, and some very, very small things. And that fluid that's formed that collects in this space is called filtrate. That, once again, precursor to urine. And then it's going to leave through the beginning of the renal tubule. What we see here is the proximal tubule. What doesn't leave the blood exits out from the corpuscle via the efferent arteriole. And so all those blood cells, all those plasma proteins should be in this blood. It shouldn't have nearly as much water, but what we're going to see is a lot of that water actually is going to end up right back in the blood pretty shortly afterwards. Okay. Now, how this all works, it's based on pressure gradients, just like what we've seen previously. Okay. The glomerulus is under a large pressure gradient, okay, because the blood inside of here has come almost not directly, but very shortly from being within the aorta. Okay. The afferent arteriole is a branch from the aorta. It's not a direct branch, but it's a branch down the line. And therefore, that blood inside of here is under higher pressure. That's going to help to force that filtrate out. Okay. And we'll get into a little bit more about this here in a little bit. But let's first talk about the renal tubule. Okay. The renal tubule is where that filtrate will flow once it leaves the corpuscle. And your renal tubule is a hollow, long, winding tubule that allows for the kind of passing of stuff back and forth between our blood and our interior, the lumen of the tubule, where eventually that filtrate, and it's almost at this point, we could call it urine, flows into a collecting duct, which is a common duct shared with other nearby nephrons. As a structure, it can be divided down into the kind of three major parts: the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. We'll go ahead and talk about collecting ducts as well down at the bottom. Here's a much more sophisticated version of a nephron. Here's a much more detailed picture than what we saw a few slides ago. So we see here in this kind of center here, the circular structure called the corpuscle. Here's our afferent arteriole bringing blood in. Now we can see those blood capillaries that have a fancy name called glomerulus. Here's the efferent arteriole draining blood out. Okay. What is not leaving the blood, or what I should say is what does leave the blood, forms in the filtrate, which then flows into the very first segment of the renal tubule, what we call the proximal convoluted tubule, or as you'll sometimes just see it listed as the proximal tubule. We call it a convoluted tubule because it's kind of a jumbled up mess, twisting and turning. And it's what we see there in yellow, whereas what you see there in red are these peritubular capillaries that are wrapping around not only the proximal convoluted tubule, but the rest of the renal tubule, with kind of the exception over here, it starts to kind of disappear. But we definitely see the peritubular capillaries around the proximal convoluted tubule, the loop of Henle, and then the beginnings of the distal convoluted tubule. And that's important because this is where we are going to return a lot of the stuff that's inside of the yellow tube, inside the proximal convoluted tubule, to the blood. Okay. That process is called reabsorption, which is the opposite of secretion, which is where stuff leaves the blood and goes into the tubule. You know, like I said, we pull a lot of stuff out of the blood, especially water, and then give it right back. And most of that takes place immediately in that proximal convoluted tubule. Okay. This tubule is composed of simple cuboidal epithelium with numerous mitochondria, giving them a lot of energy. And there's also a prominent microvillus or brush border that faces the lumen. And we can actually see that right here. It's simple cuboidal, so one layer of cube-shaped cells, but more importantly, the cells have those little finger-like extensions, just like those absorptive cells that we talked about the other day in your small intestine. Okay. And that provides a great deal of surface area, enabling a lot of contact between the cells and what's on the inside of the tubule, inside that lumen where I have the cursor now. Okay. The location of the proximal convoluted tubule is strictly out in what's called the renal cortex, which is the kind of thin outer shell of our kidneys, and is shown here in this kind of yellow area in our picture. Okay. What's not down or out there in the renal cortex, because it dips down into the center of a kidney, is this, what we call a nephron loop, which is the exact same thing as what's called here on the previous page, the loop of Henle. A long U-shaped structure where you have cells that vary from simple squamous epithelium to simple cuboidal and to simple columnar. So you are always going to have one layer. That's what the simple term refers to. The squamous, the cuboidal, the columnar just refers to their shape. Squamous are flat, plate-like. Cuboidal are cube-shaped. Columnar, column, tall-shaped cells, rectangular-shaped cells. And no matter what the shape is, the cells are typically going to lack microvilli, so they don't have the microvilli like the cells in the proximal convoluted tubule. And now we can divide, sorry, went wrong way, divide the loop of Henle or nephron loop into kind of two major segments, the descending limb and the ascending limb. The descending limb is what carries itself down into the medulla, down into the middle parts of the kidney. Okay, so it's going down, descending. And then after performing a 180, we then carry that filtrate back up via the ascending limb. Okay. Now, it's very subtle, but there are segments in the loop of Henle that are much thicker and segments that are much thinner. Okay. Like, for example, here on the left side in the descending limb, the width of the tubule, so how far across it is very small compared to over here just on the other side where it's a little bit wider. And these thick and thin segments are very important with relation to how we form our urine and how we're processing that filtrate and forming the urine from it. Okay. The thick segments are composed of simple cuboidal epithelium and is going through active transport of salt and therefore needs extra mitochondria or for ATP production, which is what makes the cells thick. Whereas the thin segment is thin, which is going to make it really, really permeable to water. So essentially what's happening is a lot of water is moving out of this side of the loop via the descending limb and going into these blood vessels. Okay. What we're seeing across is not the movement of water, but instead the pumping of salts. And that's actually going to affect what's taking place over here. And we'll get into all that later in the lecture. Okay. After doing the 180 and coming back up and out into the cortex, we can see that all the way up to this point. Then there's a kind of twisting and turning situation, just like we saw back here, which is why we call this the distal convoluted tubule. Okay. That's the third and final segment of a nephron. Okay. Its job is to conduct the filtrate out of the nephron and into a collecting duct. And the location of the distal convoluted tubule is strictly out in the renal cortex, towards the outside of the kidneys. Okay. By focusing in my illustration to this part right here, and I'm circling it with my cursor, look at what happens between the distal convoluted tubule and the afferent arteriole. They're brushing past one another, and truly they are touching one another. In fact, if I go back to this picture, we can see that connection right here. It's labeled as a distal tubule, but it would be the distal convoluted tubule. These cells here, called the macula densa, are going to be able to communicate with these cells here of the afferent arteriole, which are known as juxtaglomerular cells. And those are all part of what we call the juxtaglomerular apparatus, okay, which allows for communication to occur between the distal convoluted tubule and the afferent arteriole in order to help maintain homeostasis of blood flow. Okay. We'll get into kind of how it all works later in the lecture. Okay. It's pretty complicated, but it's pretty amazing. And I've got another analogy, because you guys know I love analogies, that I'll kind of explain how it all works later on. From the distal convoluted tubule, that filtrate, which is at this point 99% water, I'd probably say, not quite 100%, definitely down here it will be urine, but up here close to that distal convoluted tubule, we'd still probably call it filtrate. But that filtrate's going to flow into what's called a collecting duct. And a collecting duct is not a singular structure associated with one nephron. And what I mean by that is, if you look hard, you can see kind of these branches here. Those aren't just like dead ends. There'd be a whole other nephron attached here, another one here, another one there. So in other words, a singular collecting duct is not associated with just one nephron. It's associated with many. Okay, and so it collects from multiple nephrons. Okay. Now up here, close to the distal convoluted tubule, the collecting duct has some cells. So does the distal convoluted tubule, which help to really finalize that formation of urine. Those cells are called principal cells and intercalated cells. Principal cells respond to the hormones aldosterone and antidiuretic hormone, which we've talked about at various points this semester. So hopefully those will ring a bell. Remember, the hormone aldosterone helps conserve water, which is why we call it the salt retaining hormone. Antidiuretic hormone is a hormone that will make you pee less water if you're dehydrated, doing the opposite of what a diuretic would do to you. And then intercalated cells are going to help secrete hydrogen ions. So in other words, push those hydrogen ions into our filtrate to be peed out in our urine. And therefore that helps control our blood and body pH. Okay, so that's a pretty good run through on the anatomy and a little bit of dive into the physiology of our nephrons. And for those of you who took or were in anatomy class this morning, just a double dose of what I was talking about, but that's okay. Hopefully it's really clicking in your brain, so that now we can really start to get into the meat and potatoes of the lecture, which is the production of urine. Producing urine involves those nephrons and the collecting ducts attached to those nephrons, and involves three basic processes. The first of which I've talked a lot about already is the formation of filtrate, what we call glomerular filtration, and is the first step, involving the afferent arteriole bringing blood into the capsule, where we then pull fluid out of the blood into the capsular space. Okay. And it's not just water, but it's most solutes in our blood plasma. What doesn't leave the blood comes out via the efferent arteriole, which eventually transitions and becomes those peritubular capillaries, which are all these red squiggly lines that are wrapping around proximal convoluted tubular loop of Henle, etc. And then at this point, throughout the renal tubule and into the collecting duct, we're going to bring stuff back to the blood through the process of tubular reabsorption. We've talked here lately about absorption. Absorption is something we talked a lot about in our digestive lecture, where, for example, like when we take a drink of water and that water makes its way into our stomach, small and large intestines, we pull that water through the walls of whatever that organ it is and deliver it to the bloodstream. That's what absorption is. Okay. Reabsorption is putting stuff back into the blood after it's been removed. Okay. So it's just like absorption in that we're coming into the blood, but this time it was already in the blood, so we're returning it. That's why we call it reabsorption. Okay. And when it comes to reabsorption, look how much of that water that we filtered here is reabsorbed. Okay. About 99% of the filtered water and many of the useful solutes are just put right back into the bloodstream, where we don't end up losing it as urine. Okay. The opposite of that is tubular secretion, which is just like this process back here in the corpuscle. And I say that it's just like it's because there's stuff going from the blood and into the tubule. And I love the fact that they've colored this arrow yellow, because what color is urine a lot of times? Yellow, right? And these substances are going to become part of our urine that then can be peed out. Okay. If you think back a few weeks ago, back to our blood vessels and hemodynamics lecture, we actually defined reabsorption and secretion, and that's why I say reabsorption, remember, refers to the return of substances to the bloodstream. Secretion, remember, refers to the removal of substances from the blood. Solutes in the fluid formed by nephrons and collecting ducts then drains into the ureters as urine to be excreted out of the body. The rate of urinary excretion of any solute is equal to its rate of filtration plus its rate of secretion minus its rate of reabsorption. And there's only supposed to be one period there, not two. It's not dot, 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 like it's continuing. It's just supposed to be dot. By filtering, reabsorbing, and secreting, our nephrons help maintain homeostasis of our blood's volume and our blood's composition. All right, so what we're going to do is we're going to start where it all begins with glomerular filtration, which involves the glomerular capsule, the glomerular capillaries inside, and results in the formation of filtrate. Now, in order for us to form filtrate, the water in our blood plus the very small solutes must cross a 3-layer filtration membrane from the inside of the blood vessels to the inside of the capsular space. Okay. Together, the capillaries, the glomerular capillaries, the blood vessels on the inside of the capsule, and the podocytes, which are those little cells that are yellow in our picture, that look like little bugs with one eye and little legs. Okay. Those are going to wrap around those capillaries, and together those create what's called the filtration membrane. Okay. This sandwich-like assembly permits the filtration of water and small solutes, but not the big stuff, like I said, your plasma proteins, your blood cells, and your platelets. Okay. For those substances to pass from our blood and into the capsular space, they must cross the following 3 filtration barriers. Okay. The first is a very easy barrier to get through, and it's just the pores or the fenestrations of the glomerular endothelial cells. Okay. We think back a couple weeks ago, we talked about sinusoids, which are those capillaries that look like Swiss cheese, and I showed you a picture of them. Okay. Those are not what I'm talking about here. Those are found in like our spleen and our liver. Just above those, as far as leakiness go, are these types. Okay. Let me just show you a picture. That'll make a little more sense. I'll pull up my anatomy lecture. These capillaries look like this, where the endothelial cells have a bunch of little tiny holes in them. Okay. Think of these like a strainer that you can dump a pot of boiling macaroni noodles into. Okay. The holes that you see are like the holes that you find in a strainer, small enough to allow small stuff like, the water in this case, and actually both cases, water if you're boiling noodles or water in your blood to go through, but not big enough to allow red blood cells, your platelets, your plasma proteins to go through, which are kind of like the noodles. And that's why you dump them into the strainer, so you can get rid of the water and you just have the noodles left. Okay. That's the type of capillary that a glomerulus is. Those glomerular capillaries, and we can see them down here at the bottom, are very holy. Not holy as in godlike, but a lot of holes in them. Okay. And therefore they're highly permeable to many substances. So a lot of substances pass through them, except red and white blood cells, along with other stuff. Okay. Therefore, the glomerular filtrate that's formed, that passes through there, has the same composition as our blood plasma, minus the red blood cells, white blood cells, and your proteins. Okay. If you do find red blood cells or proteins in your urine, that indicates that the hydrostatic pressure in those capillaries is excessively high, or that there's a defect in the filtration membrane. Okay. Either there's way too much pressure inside that's forcing those red blood cells out or those white blood cells or whatever out, or there's like big holes, and that's not good. Okay. Now, located throughout our glomerular capillaries are what we call mesanglial cells. They're also found in the cleft between the afferent and efferent arterioles, and they act as contractile cells that can help regulate glomerular filtration. And I can go back to this picture and point those out right there. Okay. Talk a little bit more about those, and they're also right here. We'll get into that here in a little bit. Okay. Now, that's just the first of our 3 layers. Surrounding that layer is the basement membrane of that glomerulus. Remember, capillaries have 2 layers. The endothelial layer surrounded by a basement membrane. Okay. Well, this is an acellular material composed of minute collagen fibers and proteoglycans and a glycoprotein matrix. A lot of big buzzwords that we've kind of seen before. Not that those are important to understand, but what is important is that in the matrix here, that glycoprotein matrix, it creates a negative charge, which is going to prevent the filtration, the movement of stuff that's also negatively charged. Because remember, opposites attract, but like charges don't like each other. They repel. Okay. Well, if you have a whole bunch of negatively charged proteins here, they're not going to want to go through this basement membrane because they don't like negative charges. They're already negatively charged. Okay. And then finally, those pedicels, which are the little finger-like extensions of the podocytes, and the slit membrane make up the 3rd and final layer. Okay. The podocytes contain foot-like processes known as pedicels, wrapping around the capillary, and contain filtration slits, which are these little gaps, okay, or you can see them very nicely here in this picture. Okay. That also carry a negative charge, which provides another obstacle for large molecules. Okay. A thin membrane, known as a slit membrane, extends across each filtration slit, permitting the passage of molecules having a diameter smaller than 0.006 to 0.007 microns. That is extremely, extremely, extremely small, but it still allows quite a bit out. Water, glucose, vitamins, amino acids, your very small plasma proteins, ammonia, urea, and ions. Okay. So essentially what you have, if you want to kind of think going back to my strainer analogy, it'd be like if you had a strainer and you put on the outside of that strainer, underneath that strainer, a coffee filter, and then you put another strainer on the outside of that. Okay. It's just preventing the big stuff from leaving the blood, but it's still at the same time allowing the small stuff. Does that make sense? Okay. So now that we've got the basics and the anatomical stuff out of the way, we can start getting into the actual processes involved here. The principle of filtration, which is the use of pressure to force fluids and solids through a membrane, is the same in glomerular capillaries as in capillaries elsewhere in the body. So in other words, we are always trained to go from high pressure, low pressure, down pressure gradients, just like we've seen previously. But in the case of those glomerular capillaries, the volume of fluid that we have in our renal corpuscle is much larger than in other capillaries because of three things. And the capillaries present a large surface area for filtration because they're very long and extensive. That filtration membrane, as we saw on the previous page, is very thin and porous. That makes these capillaries about 50 times more leaky or leakier than other capillaries. Not all capillaries are 50 times less leaky than these, but these are 50 times leakier than most capillaries and most other tissues. And that's because of those large fenestrations. And then the blood pressure inside of those capillaries is very high because the efferent arteriole is actually quite a bit smaller than the afferent arteriole, which causes resistance to the outflow of blood from the glomerulars to be high. Okay. It's not very visible in this image, but if you look at the size of this lumen versus this lumen, it's fairly drastic. Okay, here's a really good illustration of the difference. Look how thick this red line is versus this red line. Okay. This afferent arteriole is a lot more open and a lot more blood can flow through it than what can flow through this efferent arteriole. So in other words, it's easier to get blood into the capsule, into the capillaries, than it is to get the blood out. So it's like almost like this hose here is like a garden hose that's kinked. Okay. And therefore the pressure here is going to rise dramatically, which is good because that's going to create a pressure gradient that's going to force the water out and into the space. Okay. But it's not just two pressures, it's three pressures that affect glomerular filtration. And in that blood vessels lecture that I keep referencing back to, not the one that I just pulled up in anatomy, but the one we talked about in physiology this class, we talked about NFP, net filtration pressure, which involves these three pressures. And it's actually a mathematical thing that you have to do. It's just a simple subtraction process. Okay? But it involves GVHP, glomerular blood hydrostatic pressure. So the pressure inside of those capillaries. And that's the only one that's going to help try to force its fluid out. Okay. The other two actually oppose filtration, which is why you see those arrows pointing in towards the capillaries. And you have the capsular hydrostatic pressure, which is hydrostatic pressure exerted by the or against the filtration membrane by the fluid already in the capsular space in the renal tubule. And then blood collated osmotic pressure, which is pressure due to the presence of proteins like albumins, globulins, and fibrinogen. in your blood plasma. Now, you don't need to worry about knowing these three pressures. Don't worry about knowing the definitions. Don't really even need to worry about which ones promote and which ones oppose filtration. What I want you to know is what is net filtration pressure, which is where you take those and you do the calculation GBHP minus CHP minus BCOP. Okay. And what you're going to end up with after doing this calculation is either a positive number, like we see here, or a negative number. Okay. If that number is positive, it's going to promote filtration. If it's negative, it's going to oppose filtration. Actually, you know what? Write that down. I do want you to know that. Okay. If NFP is positive, that promotes filtration. So if NFP is positive, we form filtrate. That means stuff leaves the blood, it comes out into the space. But if this number comes out negative, that means we won't form filtrate. So negative NFP opposes filtration. Now, this is important to understand because without a positive NFP, we can't form filtration. Okay. And so let's talk about glomerular filtration rate, which refers to the amount of filtrate formed in the renal corpuscles of both kidneys each minute. So in other words, it's the amount of stuff that we're pulling out of the blood, forming into filtrate, and we're measuring it in liters or milliliters per minute. Okay. In women, it's about 105 mils per minute. In men, it's about 125 mils. Now, I use those values, but most people here in America, especially, don't really comprehend how much is a milliliter because we don't use milliliters and liters as much as we use the Americanized version of like gallons. If you add it all up, that equals about 180 liters of filtrate per day, or 45 gallons. Think about a gallon of milk. You guys all know what a gallon of milk looks like. I'm sure you all have bought a gallon of milk before. You're pulling out of your blood about 45 gallons of fluid every single day. Is anybody in this world anywhere close to peeing that much urine? I sure as heck hope not. Okay. So what does that mean? Most of that glomerular filtrate has to be returned to the blood and recycled. So it's really weird. We pull it out of the blood, then we give it back. Sounds stupid and pointless, but that's just what we do. Okay. The homeostasis of body fluids requires that our kidneys maintain a relatively constant GFR. So in other words, we want these numbers to pretty much stay the same, because if it's too high, we might be passing stuff so quickly through our renal tubules that they may be lost in our urine. So in other words, we might be peeing out too much good stuff and we aren't returning that stuff back to the body. Maybe we're getting rid of too much water, for example. If it's too low, though, nearly all filtrate is reabsorbed and we're not going to get the waste products out that we absolutely need to. Okay. Your glomerular filtration rate is directly related to those pressures that determine NFP. Or in other words, any change to net filtration pressure, NFP, affects glomerular filtration rate. Okay, so these pressures that we just talked about affect how much filtration we're forming per minute. There are three mechanisms that control glomerular filtration rate: by either adjusting our blood flow into and out of the glomerulus, or by altering the capillary surface area of the glomerular capillaries, which are therefore available for filtration. Okay. The three mechanisms include renal autoregulation, and that's not the first time we've seen that term, autoregulation. Not automatic. Auto, in this case, is meaning self. So this is how the kidneys can self-regulate. Neural regulation, that's going to involve that autonomic nervous system. So yes, we're going to be talking about that sympathetic nervous system again that we've talked about so many times this semester. And then hormones can regulate GFR, which affects how much we're pulling out of the blood. We'll start by talking about auto-regulation. Once again, we call it auto-regulation because it's the ability of the nephrons, the kidneys, to adjust their own GFR rate, their own filtration rate. And shouldn't say GFR rate because that's redundant. It's their ability to adjust their own blood flow and filtration rate without external, in other words, nervous or hormonal control, which allows them to maintain a stable GFR in spite of changes in arterial blood pressure. I should say in spite of changes, not in spite changes. There are two mechanisms of autoregulation that exist. The first is the myogenic mechanism, and the other is tubulocomarular feedback. And the myogenic mechanism is something we've actually talked about before. Okay. It occurs when stretching triggers contraction of the smooth muscle cells in the walls of the afferent arterioles. Okay. As blood pressure rises, GFR also rises because renal blood flow increases. So if your blood pressure is high, you're forcing that blood more forcefully into your kidneys. Okay. And if the blood's rushing in the kidneys more rapidly, that's going to force more stuff out of the blood. So therefore, GFR goes up if your blood pressure throughout the body goes up. Okay. But that elevated blood pressure stretches the walls of those afferent arterioles. Okay, remember, those are the blood vessels that bring the blood into the nephron. Okay. And as a response, the smooth muscle fibers in that wall of the afferent arteriole contract, which narrows that arteriole's lumen. And as a result, less blood flows into the kidneys, less blood flows into the nephrons, which reduces the GFR to its previous level. Okay. Here's what I have for a figure to demonstrate this. Okay, we have normal GFR, some disturbance occurs, driving up arterial blood pressure. Maybe we're just stressed and our blood pressure throughout the body goes up. Okay, and therefore our GFR goes up because we're forming more filtrates. Okay, but at the same time that's happening, we're going to fix it pretty quickly, because as those afferent arterioles are stretched more, a couple things happen. Mainly, just pay attention to what happens here. The muscle fibers contract more forcefully, which decreases the blood flow through them, which means GFR goes down. So if we go back to like this illustration, if we're forcing blood through really rapidly, these blood vessels stretch. And as they stretch, that triggers them to contract back to normal. And therefore less blood flows in and we form less filtrate as a result. Okay. Now the opposite of that happens when blood pressure is low and those smooth muscle cells are stretched less, allows them to relax. The afferent arterioles then dilate, which increases their GFR. Okay. This is very good at regulating and normalizing renal blood flow and GFR within seconds after change in blood pressure. So it's pretty darn quick. Okay. Tubulocomarular feedback is a feedback loop involving the macula densa, those cells that are part of the juxtaglomerular apparatus. Okay. I kind of just like to go back when I talk about this to this figure. Okay. You guys know I love analogies, I use them all semester. Okay. I always tell students this is my Barbie doll analogy, which sounds really weird, but I'm going to explain it. Okay. Barbie dolls are, pretty standard toy as kids growing up. Okay. And those are probably put together sometime, somewhere as part of an assembly line. Okay. Assembly line, if you think about it, like has a conveyor belt and the Barbie doll parts are coming down the line. Somebody's in one spot, they are taking the torso, popping the head on. Next person pops the arms on, next person pops the legs on, Next person gets the Barbie doll dressed, so on and so forth. Okay. Well, if we pretend that our nephron's an assembly line, okay, we're beginning everything right here. So this is maybe where the head's getting attached, and then eventually it's going real fast and we're attaching the other stuff. Okay. There's a point right here where we can signal back to where it all began in case things are happening too quickly. Maybe the conveyor belt's going too fast and a person's not able to stick the arm on the baby or the arm on the Barbie doll. And last thing a little girl or boy wants to do that gets the Barbie doll for Christmas or whatever is to get one without an arm. That'd probably freak them out. Okay. So an easy way to fix that is to slow the conveyor belt down. Okay. And so what happens is the cells in the distal convoluted tubule can communicate with the afferent arteriole and say, hey, slow down. Okay, I can't keep up. Okay. Those cells are these macula densa cells that literally are touching and communicating with these juxtaglomerular cells. Okay. Now, obviously, we're not making Barbie dolls, but we're making filtrate. And if that filtrate is being formed too quickly and we're flowing it through the rest of the nephron through the renal tubule, if we're moving it way too fast, less solutes are reabsorbed and return back to the blood, and therefore maybe we're losing too much water, losing too much salt, etc. Okay, so these cells in the macula densa can detect the chemicals that are not getting put back into the blood, and in response secrete chemical messengers that cause the afferent arteriole to vasoconstrict. And then they also secrete chemicals that inhibit afferent arterial dilation, which means less blood comes in, which means GFR goes down. So it's like hitting the button that slows down the conveyor belt. Does that make sense? Okay. Now, when blood pressure falls, causing GFR to be lower than normal, the opposite sequence of events occurs, although to a much lower or lesser degree. And this type of feedback is going to operate quite a bit more slowly than the myogenic mechanism, though. Okay. Over on the right is a feedback loop showing you that tubuloglomerular feedback loop, and where you've got filtration rate going up, we're forming too much filtrate, not returning enough blood or stuff back to the blood. Here's those macula densa cells communicating with the afferent arteriole. What they're going to do is cause constriction of the afferent arteriole, and that therefore decreases GFR. Notice that our control center is those actual cells of the juxtaglomerular apparatus. It's not the control center that we typically would see, which is our brain. And that's because this is auto-regulation, self-regulation without outside influence. We're able to control this within the kidneys themselves. Now, that's not the only way, once again, to help regulate GFR, because then we can actually utilize the other two mechanisms. We'll start by talking about neural. If you want to replace the word neural with nervous, that might be an easy way to remember that this is how our nervous system can regulate GFR. And hopefully, since we've hammered on the sympathetic, parasympathetic all semester long, that this is going to be pretty common sense. The neural or nervous regulation of our glomerular filtration rate is controlled by our sympathetic nervous system, by the fibers of the sympathetic nervous system that secrete norepinephrine. Well, if we just, before we even start talking and explaining things, think about what the sympathetic nervous system does to our body. It causes that fight or flight response, right? What have we talked about all semester long? Things like when you're running from a bear, which is a drastic example of the sympathetic nervous system, or just simply going for a jog. Is it important to do things like digest or make urine during those situations? No. So what do you think that the sympathetic nervous system is going to do to GFR? It's going to make it go up, or is it going to make it go down? It's going to make it go down. Okay. And it does so by way of the release of that norepinephrine, which causes vasoconstriction of arterioles in the kidneys, especially those afferent arterioles. Okay. Well, thinking like right now at rest, hopefully there's moderately low sympathetic stimulation. You're not stressed or anything, hopefully right now. So you don't have a sympathetic activity situation going on. You're more rest and digest, parasympathetic. So your afferent and your efferent arterioles are dilated, and therefore autoregulation is much more important. It prevails. Okay. But if I ask you guys to get up and run in place, move around, that would be an example of moderate sympathetic stimulation. And with that moderate sympathetic stimulation, this causes both the afferent and the efferent arterioles to constrict to the same degree. This is going to cause a slight decrease in GFR. But if I ask you guys to exercise more drastically, more vigorously, or if you're experiencing a hemorrhage, you're like bleeding, okay, what we call greater sympathetic stimulation, this is going to trigger constriction of the afferent arterioles more than the efferent arterioles. And the efferent arterioles will constrict, but not as much as the afferents. Okay. But most importantly, what happens as a result is blood flow into the capillaries is greatly decreased, which causes GFR to drop. And this is good. Once again, it's not important to be digesting your candy bar when you're running from a bear. It's also not important to be making urine. Okay. And as a result of reduced urine output, that also helps conserve our blood volume. Okay. Therefore, more blood can be distributed to our important body tissues that are doing the exercising, our skeletal muscles and our cardiac muscles. So hopefully this slide, from what you've learned this semester, makes a lot of sense. Let's talk about how our hormones can regulate glomerular filtration rates. There's two main hormones involved with regulation of GFR. We'll talk about the second one actually first that I have listed there. The second one is H1 natriuretic peptide. Okay. Remember, we talked about this hormone a few weeks ago, secreted by the cells of the atria, the upper chambers of our heart. Is going to increase the capillary surface area available for filtration by causing relaxation of the glomerular mesenglial cells, those cells that are in between the glomerular capillaries. And basically, as a result, this increases GFR. Okay. If you don't remember, atrial natriuretic peptide, we talked about it being the only one that decreases blood volume. Okay. And it does so because it makes you pee out more water. Okay. And we release this hormone when our blood volume is elevated. Okay. And so at this point, we've got like too much water in the blood. What's an easy way to get that water out of the blood? Make more filtrate, which ends up resulting in more urine. Okay. When we look at the other one, it's a lot more complicated than that, though. Okay. It involves that renin angiotensin aldosterone mechanism or system or whatever you want to call it, which happens when our systemic blood pressure drops. And that results in the sympathetic nervous system signaling cells in the juxtaglomerular apparatus to secrete the enzyme renin. Renin interacts with the plasma protein angiotensinogen, causing it to release a chain of amino acids called angiotensin 1, which is then converted to angiotensin 2 in the lungs. And it's really complicated. And you've seen this figure before where you've got not only the kidneys, but the liver's involved, the lungs are involved, triggering the release of angiotensin 2, which then triggers the release of aldosterone from the adrenal cortex. That's why we call it the renin angiotensin aldosterone mechanism. So what does angiotensin do to us besides involve a whole bunch of organs? And well, we think back, it's what happens when our blood pressure drops. So what do you think our response is going to try to aim to do? Make your blood pressure go back up, right? Okay. Well, it's going to do a whole bunch of things that help make your blood pressure go back up. It's going to stimulate widespread vasoconstriction, which therefore increases systemic arterial pressure by itself. It constricts both the afferent and the efferent arterioles, which reduces GFR, and therefore we lose less water. So that makes our blood volume go up, which therefore makes your blood pressure go up. It stimulates sodium chloride, which we commonly would call salt, and water reabsorption. So in other words, we pee out less water and salt. And in doing so, that increases our blood volume. That's going to take place in the proximal convoluted tubule. We'll get into that a little bit later in the lecture. The release of aldosterone is stimulated by angiotensin II. And if you don't remember, aldosterone is nicknamed the salt retaining hormone. So it promotes sodium and water retention, which therefore increases blood volume. It stimulates the secretion of antidiuretic hormone, which also promotes water reabsorption, which increases blood volume. And it stimulates you to be thirsty and encourages you to take a drink, which therefore hopefully will allow you to drink or cause you to take a drink, which increases blood volume. All these increases in blood volume increase your blood pressure, and we've talked about that before. As far as GFR, though, this is what it's going to do. It's going to reduce the GFR. Therefore, it makes us pee less water out, and in doing so, blood volume goes up. Therefore, blood pressure goes up. So, to summarize, 26 or table 26.2 lists off the different modes of regulating our filtration rate with the two mechanisms of autoregulation. Then the neuroregulation stuff that we talked about with sympathetic activity, and then those two hormones that we just talked about. Notice everything is going to trigger a decrease as far as the table goes. This is how it's listed. The only one that increases GFR is HRO natriuretic peptide. Now keep in mind, that's just the first process involved with forming urine. If I go back to this slide, we said that there's three basic processes in the formation of urine. That's what we just got done talking about, just the very beginning. Forming the filtrate. Okay. That all takes place in the corpuscle. Then we have to move that filtrate through the long winding tubule, through the proximal convoluted tubule, through the loop of Finlay, through the distal convoluted tubule, eventually in the collecting ducts. And throughout that long winding tubule, we're going to pass stuff back and forth. Some stuff is going to leave the tubule going into the blood. That's what reabsorption is. Some stuff's going to leave the blood entering the tubule, and that's what secretion is. Okay. Let's begin by talking about reabsorption. Okay. The process of reclaiming water and solutes from the tubular fluid, returning them to the blood. This occurs throughout the entirety of the renal tubule, through the proximal convoluted tubule, the loop of Finlay, the distal convoluted tubule, and even the collecting ducts. Normally, about 99% of that filtered water is reabsorbed in these structures. Think about that. We pull out gallons and gallons and gallons and gallons of water and stuff. And then we give 99% of it, as far as the water goes, right back. And once again, I say it sounds stupid, and it kind of really is. Why we do it. It just is the way we do it. Okay. As far as the largest contribution, it's the proximal convoluted tubule that does the most as far as returning of that water. And most of our water and solutes are recovered by our blood via two routes, what we call the transcellular route and the paracellular route. And you don't remember, the prefix trans means across or through. And that's why I bolded, and I believe you have to fill in this blank, through is what we're going to focus on here. The transcellular route is where the substances pass through the cytoplasm and out the basolateral membrane of the epithelial cells that line the tube. We call this transcellular reabsorption. Whereas in paracellular reabsorption, it's between the epithelial cells that line the tubule, and we call it paracellular reabsorption. Okay. We can see both examples on the next page here in our top figure with sodium. And here we see para, here we see trans. And what we're looking at in this illustration is kind of like some of those figures we saw the other day in our digestive lecture. But here's our tubule lumen. So this is the inside of the tubule. This is shaded yellow, which is fancy, and I love it because that's fluid. It's filtrate. It's not urine, but it's going to be urine. And urine, once again, is kind of yellow colored. Here's the tubule cells that form the tube. Here's the capillaries that surround them. Okay. If the substance, in this case sodium, is going between two cells, we call it paracellular. Whereas if it's going actually through the cell, we call it trans. Now, sodium is very important because reabsorption of other stuff is primarily coupled to its movement. Okay. We are actively transporting sodium out of the tubular cell and into the into the surrounding interstitial fluid and blood plasma by the sodium potassium ATPase pump. Okay. It's located right here in what we call the basolateral membrane of our tubule cells. Okay. Which means that we're always pumping sodium from where it's low to where it's high. And that's because we're using ATP and it's a pumping mechanism. Okay. Which means that this is just going to always stay really low in sodium concentration. Inside those cells, there's not going to be much sodium because we're constantly pumping it out. So what happens is sodium really wants to go from the tubule into those cells. And we see that taking place right here. Well, look who can take advantage of that. Glucose will piggyback ride on its way into the tubule, or into the tubule cell, or out of the tubule, I should say, okay, using a sodium glucose symporter, which is just going to carry both, and you can see 2 sodiums and 1 glucose into the cell. Now, this is pretty much the exact same way that glucose moves from the inside of our small intestine into our absorptive cells. Okay. We talked about that the other day. Okay. Once inside the tubule cell, glucose then leaves via glucose transporters to enter the interstitial space and then get picked up by the blood. So glucose gets pulled in the same direction as sodium. Hydrogen ions are going to be countertransported, which means they're moving in the opposite direction, which means that they're getting pushed out into the lumen of the tubule. So they're getting secreted. This is secretion, not reabsorption. Secretion is where substances are going into the tubule, okay, instead of out, okay, into the lumen. Okay. The protein involved here is called a sodium hydrogen ion antiporter. Sodium comes through into the cell, hydrogen ions come out of the cell into the tubule. And now where is this hydrogen ion concentration coming from? It's the exact same stuff we've been talking about here for the last few lectures. Talked about it in our red blood cells. We talked about it just a little bit ago in our respiratory system lecture. It's an enzyme called carbonic anhydrase that takes CO2 and combines it with water, forming carbonic acid, H2CO3, which then breaks apart to the hydrogen ions and the bicarbonate ions. Most of the bicarb gets reabsorbed by the PCT via diffusion and then puts into our bloodstream where it can act as an important pH buffer. After the hydrogen ion is secreted in the lumen of the PCT, it reacts with the filter bicarbonate, and that forms H2CO3, and then that forms back into water and CO2. So we're kind of just doing the same thing. We're moving the CO2 into the cell and then essentially combining with water, and the hydrogen ion comes out, and then we find some more bicarbonate, and so it's kind of a cycle of events taking place. Okay. But by getting the hydrogen ions out of the cell and into the lumen, once again, that is an important thing that helps us regulate our pH. because hydrogen ions are always associated with acids. Okay. Down at the bottom, in our final illustration on the page, shows you how we reabsorb solutes. Things like chloride, potassium, calcium, magnesium, urea. Okay. Their movement helps promote the movement of water. Okay. If, for example, they move into the cell that forms the tubule, water is going to chase after it. And if it moves from the cell and into the interstitial space, water is going to move there. And then if it makes its way into the blood, guess where water is going to go? Okay. Water always tries to chase and dilute, if you want to think of it this way, solutes. Okay. Remember how we talked about like you shouldn't drink salt water if you're stranded in a lifeboat? Because it'll dehydrate you. That's because water will go where the salt is, and if that's the case, the salt will be in your bloodstream. Well, you know, pretend that this like salt, some of these things like potassium and chloride actually can make salt. Okay, water is going to go where those ions are, or in this case, urea. It's not an ion, but it's a substance nonetheless. So it's just going to move to restore osmotic balance. So that's in the proximal convoluted tubule. We'll continue talking now about the loop of Henle. And you know what? I'm actually going to stop right here. I know we're 10 minutes shy, but I don't want to start this slide because it's going to take a little while to get through. And this is a good spot to stop. And we'll just kind of refresh our minds on stuff on Tuesday after the exam and then pick up right here. We got a good jump, so I'm happy with where we're at.